So here is your cell transport and action potential review. When we look at the answers to these questions, here on the right-hand side are the possible answers: diffusion, equilibrium, exocytosis, osmosis. Release waste or cell products from inside to outside the cell. As we saw with the amoeba, when you have a cell that's going to have waste inside, it's a vesicle full of waste, that cell has to fuse with the cell membrane, and when it does, its membrane, remember this is a vesicle, so this is a membrane, it's the same stuff that this membrane is made of. That membrane is going to fuse, and the material inside will leave the cell, and that the membrane of the vesicle will become part of the cell's plasma membrane again. So the material from inside, the waste, will end up on the outside. That's exocytosis. All right, and there's the word exo. So the word is exocytosis. It's C. Diffusion of water molecules. As soon as you see diffusion of water molecules, you should automatically be thinking osmosis. That's diffusion of water through a selectively permeable membrane. That's easy. That's D. Continuous movement of particles, but no overall change in concentration. Less dynamic equilibrium. So you would expect equilibrium would be the answer. B. Movement of particles from an area of higher concentration to one of lower concentration. That's a simple concept of diffusion. So these are all definitions that we've been working on for a very long time. If you haven't gotten it, you should have come to tutoring. If not, well, I don't know what to say. You should have gotten those. This should be pretty simple. The structure most responsible for maintaining cell homeostasis is the cell membrane, right? So we've been talking about that the membranes are what create compartments, and they're the ones that help to regulate what goes in and out of material. So that's the idea of homeostasis. What is the process that allows CO2 and glucose to enter the cell, to enter the plant's cell's chloroplast? CO2 and glucose enter the chloroplast. Well, the general term is going to be diffusion, and it's going to be something that it's the best answer that you have here. It's certainly not active transport. There's a couple things that you should know at this point, and that's the first is that CO2 is a gas. It's nonpolar, it's small. So CO2 just goes right through any membrane, right through the membrane. There is no. It's very permeable. These nonpolar small molecules are very permeable. So if you have a chloroplast with these inner membranes, which we'll talk about when we get back from winter break, when we look at the CO2 will just go right through the membrane. Now, when it's time for the chloroplast makes sugar, so when the sugar has to leave, when glucose has to leave, I'm not sure why they put glucose entering the chloroplast, since the sugar is made in the chloroplast. But whatever, if glucose is going into the chloroplast, in either case, it's gonna go through. It is large, so it can't be simple diffusion. It has to be a type of diffusion known as facilitated transport or facilitated diffusion, but it's still going to be diffusion. It's going to go from high to low, just like CO2. So if glucose is going in the way the problem is asking you, then it would go into the cell through facilitated diffusion. All right. The best answer is diffusion here. Which of the following is not a form of passive transport? Oh, by the way, let's go through and say it's not osmosis because that's the diffusion of water. Neither one of these two molecules are water. It's not active transport because CO2 does not require any kind of transporter. Glucose could be actively transported, but it isn't. It goes from high to low under normal circumstances. Low to high is active transport, right? When you're going from low to high, it's active transport. So diffusion is the word that is the best, is the choice, is the best option here. Which of the following is not a form of passive transport? Molecules are too small. Diffusion certainly is a passive transport, meaning it's passive, not active, meaning you're not using ATP. Molecules are too large. Yeah, molecules that are too large do need to do active transport. So that's not going to be passive. So certainly this is something I'd like to think about. I may not choose it, but I'd like to think about it. Osmosis is passive. So I know osmosis is passive and does not require ATP. I know diffusion does not require ATP. And I know molecules that are small like CO2, they're going to do diffusion, so it's not that one. So again, the best answer is this answer. Molecules that are too large need to do active transport. It's not a form of passive transport. Not happy with the way they phrase the questions, but I think it's key that you be able to answer a question even when it's badly written and you see molecules are too large are a form of passive transport. Active transport, not passive. Diffusion continues until equilibrium is reached, Tuger pressure is reached, one side has more. Well, there's certainly not one side has more, because as soon as one side has more, then it's going to go in the opposite direction. If it's diffusion, which means it can pass the membrane easily. If it's Tuger pressure, that has to do with the pressure. Remember, I just want to remind you what Tuger pressure is, that Tuger pressure is when you have a plant cell wall and then you have a membrane inside that cell wall. As it pulls out, as it gets filled with water, that creates a pressure against the cell wall. The membrane pushes up against that cell wall. And that pushing is called Tuger pressure. It's what allows the plant not to wilt, but to stand straight up. All right. Diffusion continues until Tuger pressure is reached, so that Tuger pressure is not the answer. Equilibrium is reached is the answer. If a cell is placed in salt water, water leaves the cell by osmosis. Water diffusing from high to low is going to be osmosis. It is diffusion in a way, but it's a special type of diffusion called osmosis. Not active transport because ATP is not being used. And phagocytosis is how you move very large particles, like whole single-celled organisms even, from the outside. Remember, phagocytosis is when the cell literally surrounds it and pinches off and creates a vesicle inside the cell. Okay? A cell moves particles from a region of lesser concentration to a region of greater concentration. So low to high, low to high. And we call that active transport. Passive transport is not a thing when you're going from low to high. Osmosis does not go from low to high, and facilitated diffusion does not go from low to high. These are all formed, obviously this is all passive, including passive, right? Remembering facilitated diffusion is when you need a helper, a carrier. In the case of glucose, you need some kind of transporter protein, a channel, if you will. Energy for active transport comes from the energy for active transport comes from cellular respiration. Cellular respiration makes ATP. Now, this was not something I would have expected you to know. If you didn't know it, I'd understand it. They did mention in the previous chapters. Photosynthesis is what it captures energy and put into glucose, into sugar, and glucose. Ultimately, osmosis is not a form does not provide energy for active transport, and exercise uses energy, doesn't make energy. So. Uh, Again, another kind of not great question, but the best answer is cellular respiration. Use pictures on the left to answer the questions on the right, or so you use the picture on the left to answer the questions on the right. So without even looking at the questions, I see this is high. These glucose molecules are high on this side. How do I know they're glucose molecules? It says glucose molecules there in the blood, and here they're low. All right, so which side has higher concentration of glucose? That would be the blood. Which way will the glucose go into cell? If it can. Does this require energy? No, it's passive, it's high to low. Is this active or passive transport? It's passive. Specifically, it's a type of passive transport called facilitated diffusion. Facilitated diffusion. What specific type of process is this? Well, specifically, it's facilitated diffusion. Which way will the water go in this picture on the left? If you have a plant cell after not being watered recently, so you have to remember, you have to think about it. You have a plant, has roots, has a leaf, and you have water. There's less water on the outside than you would normally expect, and inside there's more water. So water is going to go from inside to out, right? Because there's a higher concentration of water inside the cell at this point than there was on the outside of the cell. So the plant loses water and starts to droop. So, in which way will the water go? Into the vacuole or out of the vacuole? Out, because there's less water outside than in. So the concentration gradient is high outside and low. High inside and low outside. So it's high to low, and it's passive, etc. for all the vacuoles. By what process will the water move? Water moves through osmosis. Does turgor pressure increase or decrease? It decreases. Because remember, turgor pressure is the pressure of the water pushing out against the sides of the chamber. What will more likely occur to the cell if this continues? Plasmolysis. The cell is going to go through plasmolysis, which, remember, that means that you have a cell, it has a membrane. As it loses water, that cell's membrane starts to shrink because water's leaving. Hence the idea that there's less Tuger pressure. There's less Tuger pressure because the cell is shrinking because there's less water inside. Less water inside. There's less. As the cell shrinks, it stops being able to function and it dies. We call that shrinking plasma. And the extreme shrinking, we call it plasmolysis. Plasmolysis. All right, let's go to the next one. Amoeba engulfs a food particle. Does this require energy? Absolutely. And you should know that. If you remember the amoeba that we looked at on screen, the amoeba, it takes a lot of energy to move around that and create a vacuole and then digest it, right? So yes, absolutely. Is this active, passive? It's active. Does the endocytosis or exocytosis? This is endo. Why is it endo? Because it's engulfing, which means it's taking it in. And amoeba expels waste. So again, looking at the amoeba getting rid of food, it was here, and it noticed that the membrane is fused. This was a vesicle. It had leftovers. This was left over after digestion, or maybe it's waste that was put into this vacuole. Notice the vacuole has a membrane of its own. It's starting to fuse with the plasma membrane, the cell's outer membrane. And now here it's fused. In this one, it's fused already. It's become part of the cell's membrane. You see, it's continuous. And what was inside the vacuole is now out of the cell. So does this require energy? Absolutely. It requires a lot of energy. Is this active or passive? It's active. And what kind of cytosis? It's exocytosis. So look at the black dots represent sugar, solute, molecules dissolved in water. So this is a real easy question, right? I mean, at this point, what is there really to discuss? It says in which of these is the concentration of the solute the greatest. I would really hope that no one says anything but A. And look at the diagrams of black dots represent solute molecules dissolved in water. Which is the concentration solvent the greatest, A or B? Again, it's just a repeat of the same question for some reason. I don't know why. They probably meant the least. One of them being the least, the other the largest. Oh no, concentration of water, I see. My bad. Well, the concentration of water is actually going to be highest here, right? We've done this before because there's only two things, either sugar or water. If sugar is higher, then water has to be lower, and if sugar is lower, then water has to be higher, right? So that should be that. Here, cellular transport review. You have hypo. If you are active, you are using energy. If you're passive, you're not using energy, right? And I don't know why they put hippo, but it should be hypo, right? The cell will swell, right? If you're passive, you are not using energy. And remember, salt sucks. Water will always move to where there is more solute. Oh, that's interesting. Salt sucks. It's funny. Although there's no such thing as suction in nature, but that's fine. All right, so water will move to where the salt is highest, the hypertonic solution side. So we know that. So, all right, release of waste or cell products from inside to outside. We already talked about that. That's exocytosis. That's C. Diffusion of water molecules through a selectively permeable membrane. We've already said that's osmosis. D. Loss of water from cell resulting in a drop of Tuger pressure. Plasma membrane detaches from the cell wall. We call that plasmolysis. We talked about that. So that's E. Continuous movement of particles, but no overall change in concentration. We talked about that. That's B. And movement of particles from an area of higher concentration to lower concentration is diffusion A. So those are actually repeated from the previous page. A solution in which the concentration of dissolved substance is lower than the concentration of so we're either making it true or false here, right? Solution of concentration of dissolved substance is lower than the concentration inside the cell is hypertonic. Lower, right? Lower is not hyper. Remember, hyper means high. So it's not hypertonic. So that's false. Internal pressure of a plant cell is called Tuger pressure. I can accept that. It's a little loose definition, but okay. The passive transport, the measure, the movement of particles across the membrane requires energy. And passive transport, movement of particles across the membrane requires energy. It does, but no extra energy, so I'm going to go with false, right? Just the energy of the heat energy, its own kinetic energy is enough. Endocytosis is a process by which a cell membrane surrounds and takes in material from the environment. Yeah, that's correct. That's true. A passive transport of material across the membrane by means of transport proteins is called activated diffusion. Well, that's false. That's not something we've ever used. It's facilitated. Membrane that allows only some materials to pass through shows selective permeability. That's true. Structure most responsible for maintaining cell homeostasis is the cell membrane, which we've already answered. The cell membrane is made up of a lipid bilayer. There's no layer of cholesterol, there's no layer of enzyme, and there's no protein layer. There are proteins in cholesterol and sometimes enzymes in the membrane. That's true, but they're not. There's no layer of them. So that's what makes these wrong. Okay, it's no layer. There's no layer, no layer, no layer. Which of the following is not a form of passive transport? Endocytosis, right? We talked about that. It's a lot of energy goes into that. Diffusion continues until equilibrium is reached. And then you get what's called dynamic equilibrium. There's movement up to both sides. If a cell is placed in salt water, water leaves a cell by osmosis. That's the process of water leaving. If it is not water, the tulip plant wilts because it loses Tuger pressure. We just described that, talked about it. Water leaves, the water's not pushing on the inside of the cell wall collapses. The cell moves particles from the region of lesser concentration to a region of greater concentration by active transport. It's going from low, right, from low to high. So low to high means active. What organelle produces the energy required for active transport? Well, we're going to say mitochondria, right? You don't have to know that for the test. One snuck by me. The organelle plays a large role in exocytosis by packaging proteins into vesicles. We'll call that the Golgi body. If a cell normally contains 70% solute inside, from the outside has 60 solute, what happens? All right, so now this depends, as we discussed in the past. This is one of those that depends. This is kind of the simple answer they want, is that, so it depends, is the solute permeable to the membrane or not? If the solute's permeable to the membrane, then the solute will go from inside, where it's 70%, to outside. where it's 60%. Or if the solute isn't able to go, the water will move from outside where it's higher concentration to the hypertonic side, which is inside. So it depends. One of the two will happen. When molecules move with the concentration gradient, it means that they are moving from high to low. Define equilibrium, movement of particles in both directions equally. All right. All right. So this one again is the same as the previous question, where you have high blood glucose and you have low glucose in the cell, and asks you which is the higher side. It's in the blood. Which way will the glucose go into the cell? Again, assuming the glucose can move through facilitated channel proteins. Does it require energy? No. And is this active or passive? It's passive, and specifically it's facilitated diffusion. So if you want, you can rewind this video a little bit and look at the same question on a previous page. This is also, this next one's also the same page. It says water rushes into plant cell vacuole. Is this diffusion or osmosis? Even though it's a similar picture, it's a different set of questions. So let's go ahead and answer those. It says water rushing. What kind of, is this diffusion or osmosis? Of course, it's osmosis. So is this passive or active? Transport's passive. What is the force called that causes water to rush into the? It's called hydrostatic pressure. That term hydrostatic pressure is not going to be on the test, but it is important that you just have some idea of what's going on. If you go to this website, any website, you can take a look at what happens when water rushes in. As water rushes in, it causes a pressure inside pushing out, and that's why this thing swells up and ultimately could burst. So you should be aware that is possible, that it could happen. And the same when it leaves out, hydrostatic pressure is actually reduced inside so much that the cell shrinks up and shrivels up. So, it's important that you have a vocabulary word, but it's not going to be tested in my class. Hydro for water, static, meaning standing still, pressure. So the pressure of water is called hydrostatic pressure. Does Tuger pressure increase or decrease? Tuger pressure is specifically the pressure of the hydrostatic pressure on the cell wall, right? The water pressure on the cell wall as water's going in. Now here it says water rushes into the plant cell, so now we have the Tuger pressures increasing. I think if you rewind this and look at the picture, the previous page, and look at the cells here versus the cells there, I think you'd agree that obviously this is pushing up against the cell wall more than the previous picture. Is the plant cell hypertonic, hypotonic, or isotonic? Is the plant cell in a hypertonic, hypotonic, or isotonic environment? So are these cells in an environment that has higher salt? If it was, these would shrink, they wouldn't swell. So it's not in a hypertonic. So if it were in a hypertonic environment, then these would shrink. If it were in an isotonic environment, they wouldn't increase at all. So the only way to get water through osmosis to rush in is if they're in a hypotonic solution. So the word here is going to be hypotonic. What will more likely occur to the cell if this continues? What will more likely occur in the cell if this continues? Plasmolysis or cytolysis? Uh, Cytolysis, is likely the not plasmolysis. Because remember, plasmolysis is the shrinking of the cell, so it's not going to shrink; it's going to swell up. Very difficult to happen in plant cells, but yeah, it's possible. All right, let's move on. Which term is the correct description? Has match the term with the correct description. So the description is transport protein that provides a tube-like opening in the plasma membrane through which particles can diffuse. We call those channel proteins. So that would be H. Is used during active transport but not passive transport. That would be a carrier protein, right? It could be exocytosis or endocytosis, but since they're saying is used, I'm going to go with carrier protein. Process by which a cell takes in material, and by the way, carrier protein, think about the sodium, potassium, ATPase pump, right? Think about that when you're thinking about a carrier protein. Process by which a cell takes material by forming a vacuole around it. Takes in material, that's endocytosis. Let's see, particle movement from an area of higher concentration to an area of lower concentration. We call that diffusion. Well, in this case, do they have diffusion? They don't have diffusion, but they do have facilitated diffusion, so I'll go with that one. Process by which the cell expels waste from the vacuole, we're going to call that exocytosis, that's F. Form of passive transport that uses transport proteins, again, we're going to go with B. Particles move, I kind of want to change this B to passive transport, just because even though it's possible that you could put B, we're using B down there, so if I were asked to do this for a grade, I'd probably want to use passive transport here as D. Particle movement from an area of lower concentration to the area of higher concentration. There we have to talk about E, active transport. Transport protein that changes shape when a particle binds with it. Transport protein that changes shape when a particle binds to it. Again, there we'd have to use G again. All right, so there's two G's, it looks like. So again, when you're talking about a carrier protein, like a sodium potassium ATPase pump, it usually is to do some form of active transport quick from low to high. And remember, they change shape, and that's how they get to carry the molecules across the membrane. All right, moving on. Summary of the different kinds of transport. Diffusion is passive, no ATP. I'm just going to go P or A. I'm not going to write passive and active repeatedly. P for passive. Osmosis is passive. Facilitated is passive. Active transport is obviously active. Endo and exocytosis is active. They require ATP. No, 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 yes, yes. Transport direction down or against the concentration gradient. Transport direction down or against the concentration gradient. It goes down, so down meaning it goes from high to low, down, down, against, against. Uses transport proteins, yes or no? No, no. Although this might, to facilitate diffusion using aquaporin, but we'll move on. Yes, it does use a protein. Even though it's diffusion, even though it's passive, it does use a protein. Yes, and active. And uses transport protein, no, This is the whole membrane surrounds and changes. All right, let's move on. So which of the following statements describes the conditions in the living room, in a living neuron? The sodium concentration is higher outside, the potassium concentration is higher inside. That's what we know. That's what you've been told. It was important that you know it. I told you that. Sodium concentration is higher outside the neuron, potassium concentration is higher inside the neuron. That looks a little bit, let me reduce the size of that. So again, sodium concentration is higher outside the neuron and potassium concentration is higher inside the neuron. That should be understood. That's how we start. Which of the following statements describes the condition of a living neuron? Inside neuron has a negative electrical potential. So by the way, just to be clear, this is the chemical gradient, where you have sodium, this is outside, this is inside. Here you have sodium is high, and as you go more, as you move to an area as you move into the cell, it's lower and lower. Here inside, potassium is high, and as you move out, it gets lower and lower. So that's the chemical gradient, right? Now, when you're talking about the neuron, this number two, when you're talking about the electrical potential, now this is asking about, this is asking about the electrical potential. It says inside the neuron, it has a negative electrical potential relative to the outside at all times. Well, we know that's not true. This whole point of the action potential is it's going to change. I'll go through that one more time. In fact, we'll go through that in number 5. Inside the neuron, it has a positive electric potential. No, it has a negative electric potential. We know that. It's negative 70 specifically. The electric potential in the neuron at rest is negative but becomes positive when excited. That's true, but we're going to put a little asterisk by it because I have to read all the questions. Electrical potential in the neuron of a neuron at rest is positive but becomes negative when excited. Well, that's not true. It's negative and becomes more positive. And we'll look at it in a second. Inside neuron it is always electrically neutral, which is obviously not correct. Otherwise, you wouldn't have an electrical gradient. So the answer, at least I hope it's obvious to you at this point, the answer is C. So the electrical potential, when we look at the membrane, oops. We know that inside it's at negative 70 millivolts as compared to outside. So this is a positive overall, and this is more and more negative as you go inside. So you go from very positive to very negative. All right, so a lot of positive charge on this side, a lot less on the other side. Again, this being outside and this being inside. And this again is the electrical gradient. The differences between inside and outside, there's 2 gradients, electrical and a chemical. That's why we call it the electrochemical gradient. Is which ion is critical to trigger the release of the neurotransmitters, right? And that was a question that was on your test. I didn't mean to put it on your homework, but I did. Trigger the release of neurotransmitters, and that's calcium. And I'm going to go through that in a second, but that would be calcium. I know that what we want to say is sodium or potassium, because those are the 2 that we talk about when we talk about an action potential, which we'll talk about down here. But it really is calcium that is going to trigger the release of neurotransmitters. If you did the ecstasy case study, you'd know that, and if you paid attention, or if you wrote it down when we covered it in class, pay attention, wrote it down, then you know it as well. But anyways, I'll go over it one more time here. Then when the neurotransmitter is released, the presynaptic terminal of the neurotransmitter receptor on the postsynaptic neuron will cause the neuron to depolarize, right? So let's go through the possibilities. Hyperpolarize, that doesn't make sense, right? So that's not a thing. So it's going to open up the channels and let things out. Not change the membrane potential. Well, in that case, nothing happens. So why release a neurotransmitter, right? The whole point is why. What is the neurotransmitter doing? It's sending the signal. It's transmitting the signal from one neuron to the other. It's the ligand that's binding to the cell to open up the channels to start the depolarization, right? It depends on what type of receptor and neurotransmitters are involved. This is not true. It doesn't depend on it. But there is an interesting subtext here. It's an interesting little side note. And that's that neurotransmitters, there are these things called inhibitory neurotransmitters. They're not going to be on the test, but you might want to know about them. Inhibitory neurotransmitters, an example of that would be GABA. So GABA, what it does is it blocks the signal. It blocks the receptors from other and doesn't allow the other neurotransmitters to bond to the receptor. So if you have a receptor on one cell that has a neurotransmitter that's going to excite or start a depolarization, GABA comes in and blocks it, right? So it doesn't let it happen. You have neurons and neurotransmitters that, under the right conditions, your body will send out GABA to inhibit that particular pathway from being excited. So imagine you're in church and you want to curse. You have the ability, your brain has the ability, assuming your brain is matured and is developed correctly, your brain has the ability to release GABA and say, no, don't say it. We're going to stop ourselves from saying it. The way we stop certain reactions from happening is by releasing a neurotransmitter that's called the inhibitory neurotransmitter. So that's just an example. I hope it's one that I just kind of put together, so I hope it's fairly accurate. But GABA would be an example of something that's going to stop. It doesn't. It's not that it doesn't do the opposite. It doesn't hyperpolarize it. It doesn't. But it doesn't depolarize it either. So it blocks it from depolarizing. So that is true. But for the most part, a neurotransmitter is going to depolarize the membrane. Okay, so the right answer here is depolarize. All right. So now the action potential. Let me go ahead and draw it out. It starts at -70. Starts at -70. That's rest. Then it might go. Let me zoom in a little more. So it's going to start at -70. You might get enough input where it's just not quite enough to get to -50. But you have to understand that this is an all or nothing event. Either you get an action potential or you have no action potential. There's no in between. So this is going to be all or nothing. So if you have enough stimulus to kind of almost get there, you don't even register it. It doesn't set off the axon. No signal. But if you have enough input where it gets to -50, and -50 is what we call the threshold. That's where that line at this point is when the action potential takes off. So it takes off, and it's going to get to about +30. And that's the inside of the membrane going from negative 70 all the way to plus 30. At that point, I'm going to describe what happens, but there's a change, and that repolarizes the membrane, and it gets repolarized way more than it needs to, so it goes way below where it started, negative 90 or something like that. Now, just to remind you that it started about here, right? And it's going to go below that. It's going to go way lower. And then it's going to go way lower, and then it's going to come back up and go back to resting potential. Stay there until the next signal. So this is known as the resting potential. That's the voltage difference from two sides when the neuron's at rest. Then at this point, I'm going to label these and then describe them over here. I'm just going to label them and describe them off to the side. Label this A. Label this as A. At A, what's happening, and it happens again, it happened here, and it happened here again, right? You're getting signal. You're getting some kind of signal. Now, what signal are you getting? Remember, we talked about this. It could be pressure on the type of protein protein channel that opens because of pressure. It could be light in the case of the sensory neurons in your retina. It could be ligand binding in the case of a neurotransmitter. So let's go ahead and look at the ligand binding real quick. I'll zoom out for a second. So you have this neuron that comes out from somewhere in your brain or some other part of your nervous system, and then you have this dendrite, right? This is a dendrite. This is the end of the axon and the beginning of another dendrite. This is the cell body of the next neuron. So to remember that when you're dealing with neurons, you have these cell bodies that kind of, these membranes that go out and kind of, that's a bad drawing, so let me redraw that. So these neurons have extensions. They extend their membranes out to kind of reach out and try to meet up with other cells so that they can connect, make connections. Those connections are what we call, that's how you think. You're able to think. And this is, of course, your axon of one cell. This is the axon. And then in between, you have the synapse, the place where the two meet and share signals. Now, in this presynaptic neuron, before the synapse, you have vesicles, membranes created by this cell that have neurotransmitters inside them. They're in the vesicles. They're not doing anything. They're just sitting there waiting. They're in reserve. So they're made by the cell, by this neuron over here, back in the cell body, and then they're just held here, just staying there, waiting. Waiting for what? For an electrical potential to come up to change, for the volts to change, right? For the electricity to flow, for the voltage gated channels to open. And as soon as sodium starts rushing in, making that inside more positive, then the voltage across the membrane changes, and that opens other channels. And the voltage gated channels here open. When they change, what happens is that there's these channels over here that along these channels are going to open up because the sodium is rushing in, because the membrane is becoming more positive, it changes the shape of the protein and allows now calcium to come in. Calcium has 2 positive charges for every 1 calcium, and they rush in, and all these calcium ions here, they signal these vesicles to come and fuse with this membrane, do exocytosis. So they're going to do exocytosis. So they fuse, and as they start to fuse, and they start to release the neurotransmitters into the synaptic gap, the synapse here. And the membrane that used to be part of the vesicle becomes part of the plasma membrane. And I shouldn't have made those red. I should have made them a different color. My apologies. Let's make them green. Just so we don't get the colors confused. This is not sodium, this is neurotransmitter. So it could be any one of the neurotransmitters you talked about in the case study. So these neurotransmitters are released into the synaptic gap. Now that's a signal. What made you release the neurotransmitter? A thought, a feeling, a touch, light. I don't know. Depends. Then here along this next cell, now this is a different cell. See, this is one cell here, and this another cell over here. Here on the next cell, there is a receptor. There's a receptor for this neurotransmitter, for this ligand. We call it a ligand. In this case, the neurotransmitter. So the ligand comes and binds to the receptor. The ligand binds to the receptor. And when that happens, this channel opens up. This channel protein opens up and allows sodium in. So this now is letting sodium come through this channel because of the neurotransmitter. And there's a bunch of them here. I'm just drawing one of them, but there's going to be a bunch of them. The more neurotransmitters released, the longer that it's in the gap, in the synapse, the more these channels open, the more sodium comes in, the more the membrane depolarizes. If it depolarizes enough, then you get the voltage-gated channels to open that we'll talk about in just a minute. All right, so then so let's draw, that's what's happening here, you see. Let's zoom in for a second. So when you look here, and you see at this point, before you got the -50, what's happening is you get some stimulus. In the case of the ligand-bound channels, the ligands are binding. If there's not enough, then there's no signal. So you have to have enough released that you get up to -50. If you depolarize the membrane, make the inside more positive from -7 to -50, then the next step happens. This is where you get excited. This is where it happens. This is where the next phase happens. This next phase is called depolarization. That's where we excite the neuron enough to do the next phase. And what's the next phase? The next phase is this. All this is happening here. The next phase is the voltage-gated channel. So using even a different color now, we have these channels here that when they open, when there's enough of stimulus, in the case of a ligand, it's a neurotransmitter. In the case of the receptors in your eye, it's light. In the case of pressure channels, it's pressure, whatever. Once you get enough, now you get these opening up, these what we call voltage gated channels open. Okay. And at that point, what happens is the sodium rushes in a great deal because these all open up a mass. So there's a mass of these that open up, and you get a big change, real fast change in membrane potential. It's called depolarization. So, and all this is drawn for you in your original notes that I printed for you and handed out to you. But here you see the first stage that's diagrammed here. The first stage that's diagrammed here is in fact this depolarization. So we can label that as sodium ions voltage gated channels open, the electrical potential becomes more positive as sodium ions rush in. Now, all that being said, remember throughout the whole membrane, all the time, during the whole process along the membrane, you have your sodium, your leaky sodium channels open. They're allowing sodium to come in all the time. That being said, these are not, even though they're open all the time, there aren't as many as the voltage gated channels, right? And then you have the leaky potassium ion channels allowing potassium out. And then you have, of course, the ATPase pump, the sodium, potassium, ATPase pump, pushing 3 sodiums out. And 2 potassiums in. So that these 3 are happening in the background. They help try to keep everything here at negative 70. But what happens when you get the ligand changing the voltage potential, or when you get pressure pushing on the pressure sensitive proteins that open up channels, or you get sound moving the hair cells and opening up channels, is you get a slight depolarization at first. If it depolarizes enough, then these massive amounts of voltage gated channels allow sodium to flow through really quickly, depolarizing the membrane quickly, getting what we call electricity, right? Changing electrical potentials. Once it gets up to about positive 30, these voltage gated channels, they stop, they close. The sodium voltage gated channels close. Then the potassium open up. Now they're late to start. The potassium voltage gated channels open up, but those are repolarizing the membrane. right? So this is here, this part here is depolarization. The inside is increasing sodium ions, increasing positive charge, right? All the time, this is happening all the time, but that's what's happening here. Here at this phase, we're going to have the voltage outside. Two things happen along in the membrane, and that's the voltage gated sodium channels close, and potassium ion voltage gated channels open. What does that do? That means potassium inside, potassium ions start to decrease. Potassium start to decrease inside, making and sodium stops flowing, so the membrane becomes more negative. All right. So the reason it's becoming more negative is that potassium's leaving. Why is it leaving? Because remember, what did we say about potassium? We said potassium's higher inside. If we open up channels and allow potassium through, it's going to go from high to low. So potassium's going to go out. Initially, when you open up the voltage-gated channels due to that stimulatory response, neurotransmitter, light, pressure, whatever, opens up the channels, and then if enough voltage change happens to negative 55, then the sodium rushes in, making the inside more positive. But now they close because they got real positive. These close up. Then the potassium voltage channels open up, and they go out. That's making this even more negative. So they keep going. It keeps going more and more negative. But then there comes a point where outside is actually much more positive. The inside's much more negative because the potassium voltage gated channels, let me see, are slow to close. So they keep letting potassium out until it's too late. It gets real low. Okay, so at this point here, these potassium gated channels are real low. And the inside is very negative. This side has your potassium is very low, and the inside of the membrane is very negative. But at the very end of this, the potassium channels are slow to close, but they finally do close. And once they close, the potassium ions decrease now outside. The potassium inside increases, bringing it back to normal. Resting. Back to resting. Well, how do you get that? How do you get it to go back to resting? Well, think about it. This potassium voltage gated channels are closed, so they're not leaving the cell anymore. The sodium voltage gated channels are closed, so sodium's not coming in, but these are still open. Sodium's coming in here. That's actually depolarizing membranes. But this potassium going out, and there's a lot more of these, the potential back down again. So we don't want it to go down. It's too low already. We're not closing these. What we are doing though is we are pumping sodium out and potassium in. And as we keep bringing 2 in and putting 3 sodium out, that brings the membrane right back up to its resting potential. And then the 3 of these working together keep it at equilibrium, keep it at -70 until the next neurotransmitter is released or the next pressure sensitive protein is excited or the next light sensitive protein is excited. It just keeps repeating itself as neuron communicates to neuron. And of course, it sends a signal down the axon. Remembering something that we mentioned earlier, which is that you have these axons have these myelin sheaths that allow the signal to jump. So you don't have to travel all the way down this very long, at the cellular level, this is a very long distance. You don't have to travel that far. Imagine if you're trying to go to Columbus, imagine instead of walking to Columbus, you took a plane, right? You hopped it, or you jumped it. So these zones here are where you have those ions being exchanged through the channels. These are called nodes, and then that's where all this stuff is happening. Here, this is a myelin sheath created by Schwann cells, but that's it. Beyond this right now, these are nodes. So these are nodes. These nodes are where you get the ions exchange. This is where you get the action potential happening. Of course, the whole thing is known as an axon. All right, so at the nodes, so now the electrical potential, the electricity, is going to jump from one node to another node, all the way down, rather than opening up a million voltage gated channels. You only have to open up the voltage gated channels here. So the axon sends the signal down to the next neuron. And that's how it works. Okay, so just ignore these two. They're good questions, but I think they're just more complicated than we really need to get into. It's not really that much more complicated, but we're really running out of time. And let's not talk about this one either, nor about this one. All right. I just would rather not talk about it. They're interesting questions. They're not worth our time. They are worth our time, just not worth our time. Not worth the amount of time that it would take, considering how little time we have left. So we look at a sodium potassium pump. That asks you, evolutionarily, why does it make sense that sodium potassium pump would have evolved? It's certainly not going to be this. It's because we put it on our food, right? That doesn't make sense, since what our ancestors probably had, although our ancestors may have put it on their food, that our ancestors back in the day would not have put it on their food before people evolved to the point where they could isolate salt, purify it, and put it on their food. And then even if you go even further back, our common ancestors that weren't human certainly wouldn't have done that. So potassium, B is interesting that the oceans have sodium chloride, so that would make sense of why sodium, right? Since the oceans are full of sodium and chloride. Potassium is positively charged. Ion has the greatest energy. That makes no sense, right? So I hope you know that makes no sense. They're just throwing words together there, I think. Potassium is the ion that is contained in bananas, and neurons look like bananas. I think that's really just obviously a throwaway answer choice. Hopefully nobody chose that one. So the only one that really makes sense is B, and that's the sodium chloride is the predominant salt in the ocean. And so you have a lot of sodium, so it makes sense that sodium-potassium pumps would have evolved. At least it explains the sodium part. So then the action potential is possible because some of the ion channels are gated by, but the only one that makes sense for the action potential, the best answer for the action potential, I hope you would agree, would be the voltage gated channel, right? The voltage gated channel, right? The rest of these, what? They're, I mean, they do, you could think of ways to work around making A, B, or D the best answer, but they're really not the best answer, since the best answer is the voltage gated channel, since that's what actually depolarizes the membrane and gives you the action potential, right? So it's the voltage gated channels that's the answer there. And then that's it. That's your test will involve all this, and good luck.